All right, good afternoon, everyone. I couldn't think of a CRISPR topic to discuss today. It's perfect after lunch, it's very timely, it's very compelling, and it's probably one of those technologies that is going to be lasting, is going to be an important player, and is going to be impactful for the rest of our lifespans. So my tall order today is to tell you about the CRISPR craze, because it is crazy in so many ways, and then the implications of the CRISPR craze for the future of food science. We heard this morning it's only physicists can predict the future, so we may see if some other people can predict at least the future short term for food science. What I really want to try to do here is for the next 30 minutes try to give you everything you need to know about CRISPR. It's very ambitious. But I'm an ambitious salesman, maybe. And what I'm going to do is I'm going to give you a few minutes of the executive summary of CRISPR Cas biology. That's what we call the science underpinning CRISPR. Then I'll explain to you how this CRISPR science has been repackaged into what people now call CRISPR technology. That has led to the genome editing revolution. And then really I spend the bulk of my time discussing opportunities for food and nutrition moving forward, and also maybe educate you on how it's being used already, and maybe even how it's been used in the past. I'll finish with my take on the CRISPR craze, and then its huge impacts already and in the future on society at the grand level. So I'm not going to be obnoxious with the DNA sequences and whatnot, but this is what CRISPR is, and this is what CRISPR looks like. It's a clustered, regularly interspaced, short palindromic repeats. If you guys look at DNA all the time, you're very excited. If you don't, I'm going to give you a little bit of help. This is what you should see. DNA repeats back to back to back in a locus, okay? And they are clustered, regularly interspaced, short palindromic repeats. About 99% of people who work on CRISPR and are CRISPR experts have actually never seen a CRISPR. Okay, so far you're good. Let's move on to what CRISPR actually does. So if you only knew one thing about adaptive immunity, is that we as humans are charming, compelling, and complex. And when we get infected, our adaptive immune system heavily hinges upon the stereophysical interaction at the protein-protein level of antibodies and antigens. And it's great because we have multiple organs, multiple cell types, and regardless of your site of infection, you will have this sequence-specific, structure-specific recognition complex. What CRISPR is, in a nutshell, is the same thing as this adaptive immune system, but instead of being protein-protein interaction, it's DNA-DNA driven. And because most bacteria and archaea are monocellular, right? They don't need to care about trafficking proteins to other cells and other tissues and the like. So they can do that in one nutshell and have an antibody-antigen type interaction, but it's nucleic acid driven. That's all you need to know. And the way it works is as such. CRISPR is constituted of the CRISPR Cas system. So you have those repeat, spacer, repeat arrays. And together, flanked and affiliated and associated with CRISPR-associated genes, CRISPR-associated sequences, and the proteins they encode. And the way it works is in three major steps. One is acquisition or vaccination, one is expression or transcription, and one is interference or targeting. And essentially in the first step, you vaccinate yourself by copying and pasting. Things like snapshot, picture, Snapchat, Polaroid, drawing, depending on what age and time you come from. You can capture, by copy and paste, a piece of that viral DNA and integrate it into your CRISPR as one of those spacer elements. And when you do that, you're vaccinated. Once you're vaccinated, you need to transcribe this CRISPR array into a long pre-CRISPR RNA that will get matured and processed into what's called a small interfering RNA, kind of like RNAi in eukaryotes, also called an RNA guide, if you've read about CRISPR technology before. And that RNA guide is literally going to guide an endonuclease, a pair of scissors, a molecular scalpel, or a knife towards complementary nucleic acid sequences, and specifically and precisely cleave that complementary DNA. So essentially, it's DNA encoded, RNA mediated, DNA targeting adaptive immunity. Now, there's many different types of CRISPR Cas systems. I'm not going to bore you with the details, okay? But think of it like food. Easy for this kind of audience, right? There's all kinds of food, and they're all good or they're all bad for you. It's a question of dosage and personal preferences. The same with CRISPR. There's all kinds of CRISPR Cas systems that do all kinds of different kinds of cuts, all kinds of different enzymes. And the only two things you need to know is that A, they're diverse, and B, they're widespread. They're present in about 90% of archaea and about 50% of bacteria. Now, generally speaking, people and scientists in particular try to put things into boxes so they can classify them, use a nomenclature, give them names and pet names and so on and so forth. And there's different kinds of CRISPR Cas systems. They're split into two classes, eloquently named Class 1 and Class 2. And there's different subtypes and major types. So the type 1 is the most dominant, the type 2 is the most popular, the type 3 is the most mysterious, the type 4 nobody cares about, and type 5 is the latest. And all you need to know is that they are all DNA encoded RNA mediated nucleic acid targeting systems. They all work the same way, but how they actually work at the molecular level varies in terms of what kind of guide RNAs they have, how long or short structured they are, whether they have one or multiple effector proteins that enable them to cut and slice DNA, and very importantly, what kind of DNA cleavage they generate. So some, like type 1s, are like Pac-Man. They're going to come in, they're going to chew your DNA. Okay? Some are like Cas9. They're going to come in like a scalpel, cut DNA and generate a double-stranded break, a blunt cleavage for molecular biologists. Some are like an axe or a chop. You know, if you're in your kitchen, you're going to chop your onions, you know, like a shredder. And then others generate sticky ends, right? So overhangs at the 3' end of your double-stranded DNA break. So Pac-Man, cleavage, chopper, and sticky end. Now, I'm going to tell you today about the type 2 systems. They're the most powerful, the cutest type of CRISPR Cas system, the most portable, and the most compelling. So I'm going to tell you a little bit more about how it works so you can see how it's being used. Think of yourself as a postprandial bacterium laying down after lunch. You're very satisfying. You have your type 2 system. You have your 4 genes with the popular Cas9 here, 33 repeats, 32 spacers, and they're numbered right to left not to confuse you because that's the order in which they get added. It's an iterative vaccination card. And essentially, you're out of luck. Some fly or virus comes in, will inject its DNA into the cell. It's going to be recognized as non-self. And then you're going to scan that genome, that foreign genome, that genomic injury, that foreign genetic element for what's called a PAM, a short motif. And then you're going to copy and paste a piece of DNA right next to that motif, attach it to a new repeat. Now you have a repeat spacer unit. And that small repeat spacer unit is going to get integrated at this end of the locus as new spacer S1 or spacer number 33, and you've been vaccinated. That's the vaccination event. That's the adaptation phase. Once you're vaccinated, you transcribe this as a full-length pre-CRISPR RNA, repeat, spacer, repeat, spacer, repeat, spacer, repeat. You go to your cook in the kitchen. They're going to chop it in small pieces, and each one guide contains one unique sequence that is like an antibody-antigen driver at the nucleic acid level. That small guide RNA will surveil the cell and look for complementary nucleic acid sequences, and then come in precisely, selectively, and efficiently cut that DNA using 2 nickase domains, one RefC, one HNH, exactly 3 base pairs away from the end of that word you're looking for. Okay, that's what it does. And when it does that, it chops the DNA of the virus coming in, the virus dies, and you make it. That's all you need to know about CRISPR. DNA encoded, RNA mediated, DNA targeting, DNA cleavage. It allows bacteria to survive viral infections or plasmid exposure. 3 years ago, 3 years, 6 months, and 23 days ago, not to be precise, right? The world of molecular biology changed because a couple of papers came out that showed that you can use CRISPR as a portable technology. That's what I call at the time a molecular scalpel. And you can take that restriction enzyme, programmable restriction enzyme, to generate and target any DNA of interest you want, any sequence of interest you want, and cut it, generate a double-stranded break. It's very advantageous, programmable, specific, transferable, efficient, precise, affordable, quick, and multiplexable. You can use multiple guides, multiple enzymes, multiple cuts. Now, it's a 3-year-old technology that is still somewhat imperfect because it's only 3-year-old. The protein in question is still somewhat large and cumbersome. You still have that motif sequence, specific dependent targeting. People are concerned about on and off targeting. We'll talk about that if you want. And the only true concern that I see thus far is that not all guides are created equal. Some work better than others. Now, that CRISPR technology is being used for genome editing. So the 1-minute summary on genome editing, right? Imagine you have a scalpel or a knife or Pac-Man or whatever it is, and you cut yourself. You cut your skin somewhere, depending on what part of your body you may or you may not shave, or you may or may not cut accidentally. And then once you have a cut, the endogenous repair system are going to come in and repair that cut. And then depending on the extent of that cut, the repair mechanism will vary, and the repair outcome will be more or less visible. That's what genome editing is at the DNA level. You come in and you use CRISPR to selectively, precisely, and programmably, and by the way, affordably, generate what's called a double-stranded break. A double-stranded DNA break in your DNA. And when that DNA is cut, the cell is going to repair it. And there's different repair systems, some called non-homologous end joining, others called homology-directed repair, others macrohomology-mediated end joining, alternative end joining, and so on and so forth. And different DNA repairs will repair DNA in different outcomes. Some are very quick and dirty, like you put some dirt on it, or you put some tape on it, or duct tape on it, or you stitch it with the wrong kind of fish netting. You're going to end up with like some SNPs, or a small insertion or deletion, or you're going to go and get more surgical repair, and you're going to use essentially a template to repair that sequence, either already available in the cell or provisioned by the crazy CRISPR scientists to tell and co-opt the DNA repair pathway into how to repair it. And you can have designer mutations, designer insertions, designer deletions. And in doing so, you just rewrote the genome, the code of life, exactly at the location you intended and programmably encoded. So that's what CRISPR genome editing is. You can delete sequences, you can insert sequences, you can knock out anything you want, anywhere you want, in any way you want. This is so 2015. Okay, it's like 6 months ago, right? That was like cool, but like last year. So what the CRISPR scientists have done since then, and there's about a couple 100,000 of them, is they have taken that scissor, that molecular scalpel, and then they've taken the blades off. That's what scientists do. They do crazy things, right? Where we do that. Essentially, they can turn CRISPR into a DNA binding enzyme. And what we can do now is you can use that DNA binding enzyme and make it bind DNA anywhere you want, with such affinity that it will stop an RNA polymerase in its tracks. For you guys biochemists, you're like not believing me, but you got to trust me. This is the gospel of CRISPR, okay? So we can stop an RNA polymerase in its tracks and turn off transcription. Or you can tether it to a functional activator and turn on transcription. You can tether it to an effector domain of interest, and you can deliver any effector domain to any sequence of interest you want anywhere in your genome. You can tether it to a fluorophore and visualize DNA sequences, or you can even attach it or tether it to an acetyltransferase or methylases and rewrite the epigenome, one side at a time. And that's what we now call genome editing 2.0, is you can change the text, you can turn the lights on or off, you can turn the volume up, or you can be very quiet. You can illuminate the DNA of life, and you can rewrite the epigenome. You can do anything you want to any sequence you want in any organism you want. Ladies and gentlemen, that's the CRISPR-based genome revolution. And in the last 3 years, people have been able to port, implement, and use this technology in all the aforementioned types of organisms. This is just like the tip of the iceberg. Great and tremendous advances in medicine, in vivo and ex vivo, gene therapies, antivirals, xenotransplantations. If any of those are curious to you, let me know. I'll talk to you at the break. Food and ag, this is what I'm here today. And then the plethora of model organisms. easy, complex, prokaryotic, eukaryotic, across the tree of life. Even like CRISPR, like dinosaurs. It's like you reread or rewatch Jurassic Park, and the light of CRISPR is going to be more real than you ever thought. So some of the things that we do here in my lab, actually at NC State, just a few hours south, is we're sampling CRISPR Cas systems in nature so we can advance the toolbox. And we look at phylogenies and sizes and types of cleavage and types of activities. We use orthogonal systems, so you can use multiple tools at the same time in different ways. We validate them in vitro and in vivo, and we understand how guides are comprised and how they work and so on and so forth. So we can use all those things to what we call navigating the genome. And what we're working on right now, probably just a few months away, is you could target any sequence you want without regard of the sequence limitations you had before. So you can essentially, we're like within weeks or months of being able to target any sequence we want in any genome we want and really, literally, do anything we want with it on a scale of weeks. Not days, not weeks, not months, not years, but you can do that in a heartbeat. And you can do that multiplexibly. You can take multiple different guides, multiple different tools, multiple different Cas9s, multiple different enzymes, and then do all those things at the same time in parallel or iteratively. Now, what does it matter to foods beyond the obvious? Okay, so to date, I'm going to give you an example of series of how it's been applied in food-related bacteria. And it's a non-exhaustive list, but it's a good introduction. One example is genotyping. This is a vaccination card. I'm going to show you how it works. One is engineering or naturally generating defense against viruses or against plasmids, and the uptake and dissemination, for example, of antibiotic resistance genes. One is just, quote unquote, editing genome. One is just, quote unquote, rewriting the transcriptome. One is what I call large-scale genome remodeling. Just change the whole damn house, one room at a time. And then I'll tell you a little bit about the future of CRISPR with using CRISPRs as programmable antimicrobials. That's one of the futures of the microbiome research. That's one of the futures of CRISPR research, to say the least. So I'll give you those 6 examples back to back to back very quickly. Essentially what you have here is each line is one strain, and each box here is a unique space or sequence. And what you have here is the vaccination card of 26 vaccination events in this C. diff strain over time, from the oldest vaccination event, the first vaccination event, to the latest or most recent vaccination event. And we've used this to type bacteria like Streptomophilus in dairy cultures, Lactobacilli used in dairy cultures as probiotics, Bifidobacteria, E. coli, pathogenic or not in the big 6, Salmonella, spoilage organisms in various vegetable fermentations, gas arrived from the vaginal cavity, and last but not least, C. diff. Okay, this is like just the list of what we did the last 6 years. And essentially what you can do now is you can take C. diff sequences or isolates or samples or genomes or drafts thereof from different patients, and then you can compare and contrast and tell they all come from the same ancestor. This one's been around a long time, he's made a lot of friends, so made some bad decisions. And then, you know, those 3 guys are probably the same. They're the same at the same time. And likely those guys come from those guys. So you can go back and map, you can use those to map geographically where the isolates come from, and then compare and contrast samples derived from patients that come to a hospital. Very powerful. This is also of interest to the Department of Defense for obvious reasons. Another cool thing you can do with that is you can take a great dairy starter culture. Let's say hypothetically you work at DuPont, okay? And then you have this great starter culture that makes the best yogurt in the world, but occasionally it gets hammered by those phages. Let's say phage 1, phage 2, phage 3, phage 4. One comes from China, one from South America, one from ex-Euro, maybe England, right? And then one somewhere in South America, right? And then you, but it originates from Edinburgh, right? Anyway, so you have that system, and then you expose that strain to that phage, and then select a variant that got vaccinated. And then take that variant, expose it to another phage, and select one that got vaccinated. Take that variant, expose it to the third phage, select one that got vaccinated, and you do that again, and again, and again, and again, and again. And within about 2 weeks, you have a series of natural variants made in nature by an endogenous CRISPR Cas system that you just select for vaccination events. and you do that 2 or 3 times independently, you blend those 3 cultures, you put that into yogurt, and it's going to survive processing in China or in Edinburgh or in France or wherever you want to make your cheese, even at home. Very powerful technology. Now, the way of the future here is we've shown that you can use CRISPR and essentially just provide the CRISPR guide to a bacterium that has CRISPR already. Use its own gun, its CRISPR gun, but instead of shooting at other people, you invite it to shoot itself. It's kind of twisted, but that's what CRISPR is all about, right? And we showed that whether you target any coding, non-coding, highly expressed, lowly transcribed, right strand, wrong strand, genic, antigenic, supergenic, and so on and so forth, you have less than 1 in a million cells surviving in the population of E. coli. You kill 99.99% plus of all the cells you have. Okay? And what's cool is that it's programmable. So we can come into a population of E. coli, take 2 strains that are 99.9% identical, and choose that one difference, and use it to design a guide. And tell the cell, I'm going to kill that guy, or both, or neither. Very powerful. You can do the same and regulate that between Salmonella and E. coli, take 2 things that are about only 71% identical, and say, I'm going to kill Salmonella, I'm going to kill E. coli, I'm going to kill both. Now you have a programmable, strain-specific antibiotic. You can use that, the same approach, to target streptococci, and you can do all kinds of things. And now what you can do is you can start targeting things in the genome that are not important, non-essential genes, what we call expendable islands. You can take antibiotic resistance genes. You can take toxin encoding genes, right? You can take undesirable, non-flavorful genetic elements and cut them out of a bacteria. Just like that. Question of weeks. And if you do this using just CRISPR RNAs, and you don't target essential genes, you can naturally select things that occur at very low frequencies in large microbial populations, and find and unearth and extract and screen for natural variants that are safe. All right. Now, we've been doing this for a few years now, and if you look at this as a food scientist, right, across the food supply chain, you can see implications across typing, antimicrobials, vaccinations, or genome editing for crops, for animals, for manufacturing plants, at the product level or at the user level. And we're already working with all the people aforementioned here to use all those applications, and this is just the tip of the iceberg. Just a few years from now, maybe just a few weeks from now, maybe just a few days from now, you'll be sitting in your companies with your stakeholders being like, we got to get in. Okay? And depending on what you want to do, you can use different CRISPR Cas systems, different applications, different organisms, or different flavors or tools thereof. You can customize your own CRISPR toolbox. Now let's transition a little bit and talk about the CRISPR craze for the last 5 minutes. This is how crazy the CRISPR craze has been. Scientifically, this is the publication rates, and this is the citation rate. Within one PhD cycle, depending on where you go, within one PhD cycle, okay, the whole literature went from 27 papers total. When I gave my first talk on CRISPR, wait, you could read the whole literature in one day, depending on your appetite for CRISPR news, right? And then he went from this, 27 total, to 1 paper a month, to 1 paper a week, to 1 paper a day, to multiple papers coming out every day. That's the exponential curve of the publications. That's the exponential curve of the citation rates, tens of thousands. And this is so 2015, not even funny. Okay? And almost every week, it makes the cover of one of those magazines. We all know about the CRISPR awards, the prizes, how disruptive it is, and so on and so forth, and that's great. But the challenge is occasionally scientists overhype their own discovery. I know some of those people. So let's forget about the science for a minute. Let's look at the media. The CRISPR craze in the media is even more compelling. And we can tell the future, right? Because next week, when you all get Time magazine, this is what you're going to get. Friday's issue, the cover before July 4th weekend, by the way, which everyone's going to read on their vacation. I had nothing to do with that. Actually, maybe I did. Is the Gene Machine. Made the cover of Time. When was the last time like some kind of cool science made the cover of Time? For the right reasons. At least partially. You made the cover of The Economist, Forbes, like Google Bill Gates and CRISPR. Like CRISPR is so stupid, my car's on Twitter. It's like not even funny. Time, it's on the X-Files, NPR, women scientists in Time 100, New York Times, Washington Post, everything. CRISPR is everywhere. But much like the scientists, occasionally the media can overblow a story, right? So I have the luxury of having gone to business school. So I look at it sometimes in a business standpoint. I take my scientist and professor hat off and my media magazine reader hat off, and I put my business helmet on. And this is a slide I actually made in the summer of 2013. So 3 years ago, it's not even funny, right? But what this shows you is that in the summer of 2013, all those companies, some of which risk averse, some of which not very innovative or visionary, right, jumped on the CRISPR bandwagon. Those people knew right there and then they had to be in. 3 years ago, they knew that. They had no expertise, no IP, no core competencies, no program, nothing. And they still went in. Okay? But the smartest people in the room, right, are the people who know how to make those decisions. And you will note that some of those people are large companies. Fortune 10, some Fortune 500, some small cap, some low cap, some private, some startups, some at the VC level, and some even not-for-profit. Okay? So you have all tranches of business being hit at the same time with the same technology across different businesses: food, ag, pharma, biotech, and that never happens. For the same technology to be impactful at the same level in different kinds of businesses across all different sizes and competitive plans. That's what's compelling. And the smartest people in the room arguably are the people investing your retirement money. And they're having a tough week. Okay? And those guys, they've put in hundreds of millions of dollars in those startup companies. Hundreds of millions of dollars. Your retirement funds at work. Okay, Intellia, Editas, CRISPR-TX, Caribou Bio, and that's just the beginning. Very compelling. So in closing, this is the CRISPR craze. Just like an appetizer of the CRISPR craze. Right? You're all going to go back on new planes, or the Amtrak, or in I-95 traffic, right? And then read about how scientifically disruptive the CRISPR revolution has been. Read about the broad media coverage of the CRISPR craze, the business interest across medicine, ag, food, biotech, across business sizes and so on and so forth. But we also have to be mindful of the flip side of the equation, the other side of the coin. There are issues about IP. It's like a whole hour conversation. There's issues about ethics. There's issues about regulatory, GMO, non-GMO, and how we're going to get along and how we're going to move along. And one of the key points I want to send to the audience here, given who you are and how much of a leadership position you are, is we have an opportunity in the food and ag business to use existing, powerful, difference-making legal frameworks to decide whether they're going to accelerate or decelerate the CRISPR craze. And then whether the first business to market will be in food or ag or in medicine. Because the window of opportunity is open, and now is the time. So on that note, there's different interests in the room here. Some people are worried about breeding of plants and crops. Can we make healthier blueberries? People are working on this. Can we make healthy carrots? People are working on this. Can we make healthy chicken? Healthier probiotics? More resistant dairy cultures? Engineer the microbiome? Across food, across ag, across medicine, and across biotech, I would invite you all to think and ponder about the future. Now, I hope you will all bring the CRISPR element to your lab, your business, or your life. And understand that this is a rare, as a food scientist, I always tell the story, right? You have all the ingredients you need for a masterpiece. It's a great topic, it's a great technology, now is still the time. It's a great place, like the industry can make a big difference. It's where it came from. Ask me about that. There's some interesting, colorful people, so opinionated people working on CRISPR too. Excellent business opportunities, excellent investments. I'm not here to dispense any advice on where you should spend your money. And then also be mindful of the considerations, you know, the IP, the ethics, GMOs, and regulatory path. I hope that was a good, worthy, inspiring, and mind-opening introduction about the science of CRISPR, the technology of CRISPR, the opportunities, impact, and my perspective on CRISPR. And I will close with my conflicts of interest slide. I should have put that first, but I didn't get the memo early enough. So all those people on the left are people who joined me on that journey for the last decade. Especially when I mention my colleagues at UC Berkeley and Penn State, and Wisconsin to a lesser extent. I do get some funding, a sliver from the NSF, maybe a sliver from the NIH, and then some more from the NC Biotech Center, but I have usually received a lot more money from the smarter people in the room, right? Right? I didn't say whom. So I still get money from DuPont, not just my stock, but the actual DuPont, Pioneer, Dharmacon. For disclosure, I am chairman of the board and director of the board of Caribou Biosciences since the inception. I'm a founder and scientific advisory board member of Untitled Therapeutics, and I'm an investor and member of the ASAB and founder of Loews Biosciences. I have a lot of colleagues in the government doing a great job with CRISPR across different branches thereof, the USDA, the FDA, and even NCBI. And then last but not least, a lot of people at NC State helping me in that journey. I'll thank you for your attention. I think we have a couple of minutes for questions, and then I'll be here at the break at 3 o'clock. Thank you.
Questions for Dr. Barrangou? Thanks, Rodolphe. On your considerations slide, you might consider adding communications, because that's such a driver for the other aspects of the bullets on there about regulatory and such like, because I don't have to tell you how this is perceived by consumers and advocates and how this can be positioned, and you showed some of the headlines, could get way ahead of us before we have a chance to put it in the right perspective.
Yeah, absolutely. I mean, I could not agree more. Obviously, I'm quite mindful of it. I'm quite aware of it. I think a huge part of CRISPR's success is going to be preemptively educating the public about the technology. So actually, I'm involved in a CRISPR documentary that's being shot. We're working on the storyboard right now. It's not imminent. It's not like next week or anything. But there's people in the field proactively involved in it, because we know you have to educate the public. There's actually already some noise about like anti-CRISPR organizations being built around the world. And it's like the CRISPR craze is like the wrong way, right? Or not, just what the public thinks. So I could not agree more. It's been on a few of my slides, maybe not this particular rendition, but PR is very important. And actually, I think that an organization like ILSI or academics like myself, when I wear my academic hat, have a very important role to play to educate the public, not just educate the next generation of scientists. I don't know which one is a harder job. But really educate the public into what it is and how we can be used. When we talk about gene drives, we don't talk about gene drives because I don't do gene drives, but like gene drives, for those of you curious about that, it's very powerful, but also like liabilities thereof are very concerning. So very good point.
Excuse me, are you aware of some efforts on behalf of any of the regulatory agencies to get into the game ahead of it so that we can say, well, here are things you can do, or here are things you can't do, or here are things you shouldn't do with regard to the technology?
Yes, but not enough. Now, maybe it's never enough, right? So there's already been some regulatory statements about the status of non-regulated white button mushroom that's been CRISPR edited. So that came out. And then within a week of that was about 6 weeks ago, and within a week of that coming out of Penn State, actually, there was likewise an opinion of non-regulated organism regarding the DuPont waxy corn from Pioneer hybrid. Those have been covered in the news. This is why I'm mentioning there's an existing framework for the exploitation of this at the ag level. In terms of food supply, right? I mean, I could have asked the question, like, who's eaten any CRISPR product as of yet? Anybody? Other than me in my lab? No? So the joke is you all have. Because since 2011, a substantial portion of all dairy cultures used for yogurt manufacturing have been naturally CRISPR enhanced. So, it's a matter of how you perceive it. Do you know it? Do you not know it? Do you perceive it? Are you concerned about it? How do you do the PR? This is public information. There's like no secret coming out here, right? There's CRISPR in your yogurt. That was an article in The Atlantic and The New Yorker that came out a few years back. So yes, there's already some signs. I think the regulatory agencies have been engaged. They know of CRISPR. Everybody knows of CRISPR. But I think the level of proactivity is, at least in my mind thus far, a little bit underwhelming. And there's a tsunami coming. You can see it coming. And I'm just not sure everybody's ready as of yet. But they'll get there. I think the right people are at the table, and they're mindful of it. That's one of the reasons I'm here. One more question? Going once? Going twice? Thank you.